through the homework, we hope that you can see how the different algorithms are used and how you can string them together to really answer an important biological question. So I want to show you some scenario, and then we can brainstorm on how you might want to analyze the data. In the first scenario, let's look at this case. We know that a transcription factor X is important in some cancer Y, but so far transcription factor ChIP-seq in tumors has been very tricky. And say you have a collaborator, they generated this transcription factors ChIP-seq data in a cancer cell line, not in tumor samples, so they can grow a lot of cells. They also generated the RNA-seq data in the same cell line. When they have the factor active versus when the factor is inactive, say they do a knockout of the TF. And so they generated expression. You don't usually do ChIP-seq when the factor is inactive because you just don't really get anything. And so you have 2 RNA-seq and 1 ChIP-seq data. And so how do we go about analyzing the data? We usually start with QC and the raw, like pre-processing, right? You map the reads, look at RNA-seq quality, and you call peaks, and look at your signal to noise. Are you calling enough peaks? And make sure you get rid of batch effect in the RNA-seq. And then we can look at the comparisons, which is like more differential expression. I think I have. Yeah, sorry, I forgot to mention this. You want to look at the differential expression using DSeq. Look at what pathways or genes are enriched. You can also use the peaks to look at motifs enriched there, and then use the ChIP-seq overlap in Systrome to see what this factor might be interacting with. Then you try to integrate, for example, the ChIP-seq data with the expression, right? You can use it to refine the actual TF collaborator that you predicted. Is it GATA3 or GATA1 or GATA5, right? You can also use some public tumor data to help you with this. Maybe some factors were never expressed in that cell type. You try to combine the binding with the differential expression to infer the direct target of the TF. And then you can do gene ontology or gene set enrichment to see what this pathways are enriched. And finally, you might want to see how much of this TF or this pathway is relevant in a public tumor cohort for the same cancer type. And we will, in the last module on TCGA, we will also see whether these gene expression, high expression of these are relevant into the clinical features. But because the data was generated in here in a cell line, you want to see whether this has any clinical relevance, right? You can use that also to see in gene set enrichment, is this, sometimes it can already tell you this is related to metastasis or death risk for some disease or things like that. So now second scenario, let's say you have some disease versus normal. You don't know which factors are involved. So without further knowledge, you can look at RNA-seq, you can do a TAG-seq, or maybe K27 acetylation ChIP-seq of some normal tissues or the disease tissues. And so for that, you want to guess what factors might be involved in this disease, right? And so again, you start from QC and processing of the raw data, RNA-seq, generate RSAM, look at the clustering, make sure there's no batch effect, the disease normal are separated. You also want to do the attack-seq. Hopefully, yeah, again, usually I think attack-seq and the disease normal are very well separated. Also K27 acetylation ChIP-seq, call the peaks, make sure your experiments are working, there's enough peaks compared to the background. This next one is more interesting. You want to identify the differential peaks, you want to identify the differential genes between the disease and the normal. And also based on the differential peaks, you might be looking for motifs and also look at the peak overlap with other public ChIP-seq data and to guess what your factor is, right? What might be relevant. And then you might want to integrate the you might want to impute where this TF is binding based on the, the ChIP-seq overlap with your attack-seq. And based on the binding and the differential expression, you can infer the TF targets, you can annotate the pathways, and you can see whether this is related to the disease. You can, in this case, use K27 acetylation to look for super enhancer targets, because we say, okay, what are the really important genes for the cell? And you can ask, what genes are like a unique super enhancer in the disease state compared to the normal state? What are the upregulated genes between the normal versus the disease? What's the upregulated genes in the disease compared to the normal? And you can see, potentially the K27, if a gene is upregulated and it has a super high K27 acetylation in the disease, those genes might be super important. And you can see whether that pinpoints to the TF you are interested in. And then you might examine other public data on this disease and do gene set enrichment to see, what other public annotation you can get for your data of interest. And you might follow up by some experimental validation. If you think this gene is super important for that disease, you knock out the gene. Do you see the cell differentiating back into the normal state, or do you see the cell die, maybe in the cancer setting? To validate also the TF that you predicted are super important for the cell. The third scenario is, let's say, this is the myeloid cell. This is one blood cell lineage. They can be differentiated into, monocyte and neutrophil, so platelet, erythroid. This is red blood cell. And just different cell types. And let's just think of the case which we want to see what is involved in here. We potentially generate RNA-seq, histomark ChIP-seq, attack-seq, but let's just say we also generated Hi-C data and the DNA methylation in all the conditions. So there's a very expensive experiment. And so you might also want to start with, QC and processing of each data separately. So, like RSAM, mapping, reads, and max. So for Hi-C, you learn in the class how to kind of at least figure out what kind of interactions happen. You might be able to call some TAD domains in the data and see whether the TAD domain boundary change, and also during the differentiation. So that's kind of, more in the differential analysis to see, what's the difference between this stage and the more differentiated stage here, or basically you compare the father with the son, or you compare with the siblings. What is the difference of the adjacent conditions, right? What are the differential genes on RNA? What are the differential peaks on the ChIP-seq and attack-seq? What are the changes in the TADs? You will see that most of the TADs are really conserved, but you never know, maybe there are some small changes here and there. Do you see the compartment change? That's B TADs now becoming A, meaning that this region is from overall more silent to our overall more open regions. Also, DNA methylation. It could be that as you differentiate, new sites are being activated and then the old sites are being silent or bisulfite sequence methylated, or sorry, by DNA methylated from your bisulfite sequencing data. And so you can look at those differentials. And then next one is to integrate. How do these are associated with each other? What is the, I think you probably will see that when DNA is methylated, the histomark is the active histomark decrease and the repressive histomark increase. Usually with DNA methylation, the nearby genes are lower. And when you have the compartment that change from B to A, the histomarks are more active, the genes are more active. And so you can look at what those regions are, what those genes are, and you can look at gene ontology and pathways, right? And what are the TFs that are involved, and do gene set enrichment of the target genes. And these are probably, in the normal stage, but if you want to go even further to look at external data, you say, now that I know how the myeloid cells normally are expressed or differentiating like this, in cases where I have a leukemia or some, something that we know maybe the cell is stuck in this progenitor cell and it can't go on, you can evaluate those public expression data and say, now I understand what happened there. Some factor is not turning on, therefore the cell is not differentiating, and they keep on staying in the progenitor or the stage and proliferate. So that's the general idea of how you want to analyze data. I hope you can appreciate that. You always start from the basic processing just to generate the mapping and the pCause and the expression index. And then you try to do some differential analysis. And the third stage is you try to integrate the different data types, binding with expression with epigenetics to refine your hypothesis. And finally, you try to get some external data to better understand the data you generated yourself. Okay, that's on the second.